our talk with the work of Ladislav. So,
Thank you very much. So I'll be presenting the work on recently that I've done on modeling post-treatment gene expression with deep generative models. So this is towards our larger goal, as Anna said, to integrate multiple types of data to be able to do drug response prediction in clinic. So when a patient comes in, we want to be able to integrate the cell line experiments, also this kind of perturbations experiments that I will be talking about and so on in one model. And we believe that the right choice are probabilistic generative models that can handle all this data and subtasks that we want to do. And what I'll be talking today is just one part of it, and that's the perturbation effects. So I'll give a brief introduction into the kind of data that we use. The deep generative model that we use is based on variational autoencoders, so I'll give you a quick introduction to that. And then our perturbation variational autoencoder is relatively straightforward extension of that, and show you our results and conclude the talk. So most of the data that we have are experiments on cell lines. Cell lines are have been around for decades and are the most abundant model system to study biology of cancers and of the drug response. And there has been a lot of data generated using cell lines that measure mostly sensitivity of the cell lines to drugs when it comes to the outcome of the treatment, like how well the drug actually is efficient in killing the cancer. But a unique kind of data came from Link's connectivity map consortium, where they measured actually how these drug perturbing agents actually change the gene expression of the cell lines. So it's not really the outcome in the sense of like viability of the cell line, but what is really the impact of the drug on the gene expression in the cell line. And this is what we decided to model. So for example, for bortezomib, when you plot the t-SNE of 51 cell lines that they measured for bortezomib, you get this kind of plot where they have a bunch of biological replicates for each cell line. Like kind of skipped through that. They have relatively few cell lines that they measured for each drug, but they come from variety of tissues, and they've done a lot of biological replicates, which is good because now we can see that there's actually a lot of noise in the data, and the effect of the drug, even when we look at the highest concentration under which it was tested, is relatively low compared to the variance that we already have in the data. However, if this kind of shift is consistent, we still hope that we will be able to extract something from that kind of data. So here what I'm showing is for each cell line, all its biological replicates are in one color. So for example, here this greenish cluster, and there's a line connecting it to its post-treatment state in the same embedding, and those are all in some orangish color. So this is how roughly how the data looks like, and we want to be able to learn as much as possible from this kind of perturbation data, and in the future empower the models for drug response prediction. And as I said, the framework of choice are deep generative models based on variational autoencoders. That is recently introduced, like 3 years ago, so not so recently, a framework for inference in graphical models based on deep neural networks. And I'll show you an example how we can use the variational autoencoder for just as a latent model for gene expression. And then after a few slides, I'll show you like how we can extend it to model those perturbations. So it's a latent in its simplest form. It's just a latent model, probabilistic latent model where the joint is factorized to the prior over the latent representation, and then we have the conditional over the observed data. And the prior is usually zero mean identity covariance Gaussian, while the posterior is Gaussian distribution. But what is kind of new in this framework is that the parameters of these distributions are computed by a neural network based on the latent representation. So it's still a parametric model from that perspective, that you get a distribution over your observed variables, but that distribution is computed by a deep neural network. So that computes that the mean and the deviation, standard deviation of the distribution, and it's flexible. You can model quite complex distributions in this framework, but that always comes with costs. The more complex the graphical model is, the harder the inference usually. So in this case, of course, also doing inference and training this model is intractable directly. So the way how it's done then is using variational inference, that there's approximate posterior distribution introduced to approximate the true posterior, and this approximate distribution Q is also in similar fashion as the generative one, parameterized by another deep neural network. And then this model consists of these two distributions, of the generative one and of the approximate inference one, and we need to fit this two distributions that are parameterized by two deep neural networks to maximize the evidence of the data, the P of X. So that's still active research, how the best to do this, but the standard approach is to optimize the variational lower bound. Then that takes the form of the reconstruction error, expected reconstruction error, and some kind of you can think of it as regularization term, that is the divergence between the posterior and the prior on the posterior. So how does this works all together? I'll try to illustrate it on example. So you start with gene expression. In our case, it's we use the 1,000 landmark genes, and you run it through that approximate inference model, that is, you can also call it encoder, that in our case is a fully connected neural network with one layer of 500 hidden units and then 300 hidden units. That gives you then the parameters of the latent for the Gaussian distribution in the latent space. So that's your posterior, your approximate posterior. We use 100-dimensional Gaussian, as I mentioned, and then you sample from this posterior, and you propagate this sample through the generative process, through the decoder, trying to decode it back to the original data space. And we use mirrorly set up a neural network that first goes through 300 and then to 500 hidden units, and gives you parameters over the data distribution. And what you then, how you're fitting this model, you want to achieve the best reconstruction error between this between a sample or the mean of the reconstruction distribution when you started from some particular x. So this is a point estimate on the expected value of the reconstruction error, because we just took one sample, but using the reparameterization trick as was introduced by the original authors, this is unbiased sample, so usually one or two samples are good enough to estimate this expected value well, and then the regularization term, or the KL, between the approximate posterior and the prior on the posterior is then the KL between your posterior and the zero mean identity covariance prior. And since both of them are Gaussian, in our case, you can compute that KL numerically. so you don't have to estimate that. But if you really want to, or if you want to improve the kind of expressiveness of this model, what has been recently introduced last year are something called normalizing flows, and the particular inverse autoregressive flows that we use to further enrich the family of possible posterior distributions that you use as the variational approximation. And how does that work is that after you take the sample from the first Gaussian, you run it through this flow, which is a series of autoregressive, which is an autoregressive autoencoder basically, that further changes that sample, and that way changes the manifold of the posterior, and you can achieve better performance, slightly better performance usually. So this is a little trick that we also use. Okay, so now that was kind of the model for that was the variational autoencoder as published a couple of years ago by Kingma and Welling, and in this framework we decided to model the perturbation effects of the drugs. And what we do, we extend this latent model for gene expression by a function in the latent space that models the drug effect. A trick that worked for us well is to initialize this, or to model this function in this form where it starts as identity, and then only during the training it starts diverging from the identity. So first our drug effect function is just not doing anything, and it's better than some random initialization. And then the other thing is that both our encoders, our decoders from the latent space, and also encoders then to the latent space are shared between the pre-treatment and post-treatment state. And this way we can use also other cell line data from, let's say, cancer cell line encyclopedia and so on, to pre-train or jointly train the embedding into the latent representation. So we can actually use a couple more than 1,000 cell lines then to train the latent embedding, and then utilize those 50 or so cell lines from the Link's perturbation data to learn the drug effect function in the latent space, which is much more feasible because now by then we are working in much lower dimensional space. So then when we are inferring the posterior over the over the latent representation of the post-treatment state, we have actually two paths. Depends on if you actually have the perturbation pair or you don't. So for the training data you do, and then you have the direct path where you just encode the observed post-treatment state into the latent embedding, or you have only the pre-treatment state that you embed to latent space, and then apply the drug effect function to get the predicted latent representation of the post-treatment state. And we train then the model to match these two posteriors, and we add another KL term into the objective of the model. So this way the model is basically one-step recurrent model, and what we then tried to do is to train this on a set of 19 drugs. For each drug we trained the model separately, and as you can see, like, there's very few unique cell lines, even though there are several couple hundred biological replicates, but the underlying underlying complexity of the data is much less, because it's only from 56 or up to 56 unique cell lines. And then, as I said, we use somewhere around like 1,200 other cell lines that were not in the perturbation data set from Cancer Cell Line Encyclopedia and Genomics of Drug Sensitivity in Cancer to train also the latent representation. So we're using that kind of data to improve the encoder and decoder part. And as I said, we are using only the landmark 1,000 genes in this model. So how do we evaluate, like when we fit this model to predict the post-treatment state, how do we evaluate that it's doing something reasonable? So the way how we decided to do that is that we run this kind of this posterior of Z2 given X1, and then decode it to the data space into gene expression. So that gives you this X2 tilde, and you can compute then correlation between this predicted gene expression and the actually observed gene expression in your test set that you haven't seen before. And if I tell you that this is, I don't know, 0.6, is that good enough? Who knows? So what we wanted to then check is that if it's doing if this drug effect function is really doing something reasonable. So we compared then to correlation of just reconstructed pre-treatment state to the post-treatment, true post-treatment gene expression state. And if our correlation with the predicted one is higher than the one with just reconstructed pre-treatment state, then our drug effect function is actually moving in the latent space in the right direction. As I said, we initialize the drug effect function first as identity, and it learns this during the training. And if on our test set we see that this correlation is higher than the correlation with just reconstruction, then the drug effect function does at least something reasonable. So how are the results? We ran then ran the model in 10 times 5-fold cross-validation, and then computed Mann-Whitney test to see if really on this in this 50 experiments that are, even though not IID, to see if the desired correlation is higher than the other. And it's the case for 5 out of 8 drugs for which we have at least 51 cell lines. For those that we have less, the model doesn't really work that well. The drug effect function just overfits. And so to conclude, I guess I'm running out of time as well. So I showed you that we can use this kind of framework to model the latent space of gene expression, and then also the drug effect function that is nonlinear, because even though it's linear in the latent space, it's highly nonlinear in the actual gene expression space, because the embedding is highly nonlinear. And we can extend these models to do much more. So for example, we can then add classification on top of this model, and that's something that we call drug response variational autoencoder, where we extend it to actually classify the response of the drug. And that's something that we posted on arXiv recently. And in future, we want to extend this this model and work in this framework to do better in this prediction of treatment response. We can add and model dependency on the concentration of the drug. And since it's a generative model, we can also try to incorporate patient data or PDX data and so on, and incorporate the domain adaptation into the model. And of course, you can do multitasking model as well with straightforward extension for drugs that are similar, and one can inform the other. And that would be it. So thank you very much, and I want to acknowledge my lab and also our collaborators and all the funding that we received. Thank you very much for the attention.
Is there any questions?
Sorry, do you plan to share the method or the train methods, especially on GitHub or anywhere?
So we have to run inference on each of those, and also you need to run some kind of normalization for the data first. So we can so it's not as easy to just post it and then you like click and run inference on new kind of data. But we can then retrain the data, especially if it would be another batch. But we are not posting like a service where you could just check the outcome. But once it's published, we of course will publish also the code and so on.
So if I understand correctly, the input to your model is just the expression before treatment.
Yes.
But of course, the cell lines also have other information on data, the genomic profiles and so on that can be useful for predicting the expression after treatment. Do you have also planned to try to incorporate them?
Yeah, that's something that we can also do in the model. Depends on if we can have that kind of data also for let's say the sensitivity data sets, so that we can then leverage that type of data also to predict response, because we are mostly right now focusing on actually predicting the treatment outcome, while this is kind of the data that we want to harness in the model. So if the other data sets that would contain, I don't know, mutations and so on, then we would be look into that to incorporate it into the model. It's possible.
One small question again. So beside the dosage dimension that the treatment has, I think it also has the time after treatment. How did you collapse them into one, or how did you deal with it?
Yeah, we just for these experiments we took the highest concentration, and I think they didn't measure different times at that point. I think so. I think it's just concentration. I might be wrong, but we took just the maximal concentration. And
I think there's also a time dimension, like 6, 12, 48, 24. I think.
I see. We in that case we collapsed them all into one.
Okay, thank you.
Hi. For versional autoencoder, you have to sample from Z, right? Z. To do the decoder, you have to sample from Z.
Yes.
So if the joint distribution is quite dynamic, how do you know you sample from which point correspond to the real? How do you do it? You do the sample several times, average, and how?
So as I showed you with the kind of small illustration, like how the training works, you like when you have the posterior over in the latent space, you take several samples, but those samples are unbiased samples from the posterior. So it's not like Markov chain or something that they would correlate highly. Like so, in practice we just take 2 samples, and that's enough to get low variance estimate on the reconstruction error, and then you can backpropagate and update both the generative and the inference model.
Yeah, that's for training, but for testing.
Yeah. For testing we use for testing we use I think like 10 samples to estimate that. And then average them and then propagate that through the decoder, and then we take the mean of the predicted gene expression.
Yeah, what I mean is that if the distribution is quite dynamic, then you sample from different places of Gaussian distribution, you may get very different reconstructed signals. That might be
yeah, that's something that we also will be looking into. Yeah, that's true. Like we haven't done experiments where we would estimate that. Like how much does that vary?